Hello, everybody, and welcome to today's webinar about the challenges and opportunities of moving from preclinical to clinical stage. This webinar was collaboratively developed by GARDP and CARB-X, JPI AMR, Repair Impact Fund, Wellcome Trust, ASM, and ESCMID. It is part of the Antibiotic Bootcamps for Developers series, which have been developed by GARDP and its partners at the ASM ESCMID Conference on Drug Development to meet the challenge of antimicrobial resistance since 2017. As this year's conference could not be held, the content was developed under the umbrella of the Revive webinar series. Revive is GARDP's education and outreach program and was officially launched in 2018. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities, and all webinars are recorded in full and can be viewed after the live broadcast on our website revive.gardp.org/webinars. As always, today's presentations will be followed by a Q&A session. You can submit your questions at any time during the webinar via the questions panel, as shown on this slide. We will address the questions after the presentations, and we will do our best to respond to as many questions as possible. Our moderator today is Erin Duffy. Erin is CARB-X's Chief of R&D, and she kindly helped us to put this webinar together. Welcome, Erin. I'm now handing over to you for an introduction and to welcome our speakers. Please don't forget to switch on your webcam and your microphone.
Hello, Astrid. Thank you very much. I'm very pleased to be here today and pleased to have a conversation, or at least start the conversation among 5 of our CARB-X portfolio companies, discussing their challenges and opportunities coming from preclinical through to clinical stage. Are you controlling my slides, Astrid? Yes. Okay. Can you move to the next slide, please? Before I do that, just a few words about CARB-X. Next slide, please. So CARB-X is a global not-for-profit partnership led by Boston University that supports the development of antibiotics, vaccines, diagnostics, and other products to fight drug-resistant bacteria. We have several funders, including BARDA and the NIH from the US, Wellcome Trust and GAMRIF, or the UK government, the German government. We also are supported by the Bill and Melinda Gates Foundation. And not only do we supply support in the form of financing to companies in the fight against AMR, but also we put a significant amount of support around these companies through internal expertise, subject matter experts, and also a series of accelerators listed here. These accelerators provide both business and scientific support to our companies. Next slide, please, Astrid. So as I mentioned, CARB-X supports the development of many projects that address bacterial threats. These are therapeutics or treatment, small molecules, direct acting, indirect acting, etc., non-traditional products, including antivirulence projects and the like. We do support rapid diagnostics, as you see here, and that has been to date focused largely on pathogen ID and AST. We also support a number of prevention approaches, including vaccines, microbiome-based approaches, antibodies, and phage. Our emphasis has been on pathogens that are on either the CDC or the WHO urgent and critical lists. CARB-X begins early in the drug discovery process. So for therapeutics and prevention, that's in the hit to lead stage. And we do fund up to and including a first in human program. Where diagnostics are concerned, we start early at feasibility and take companies all the way through to verification and validation. At these points, then the programs would graduate from the portfolio and continue down the line of clinical development and ultimately towards launch. What we're talking about today is really that transition from the preclinical or the development stage through to a clinical stage program. And there are several questions that come along with this, and many of them are around how best to develop a product. What does that path look like? And how do you engage the regulators? Next slide, please, Astrid. So today we're going to hear from, it's a lot of speakers. I think, as Astrid may have said, probably more than usual in these webinars. But what we really wanted to do was give you a sense of the challenges and opportunities across the portfolio. So today, first, you're going to hear from Bala Subramanian. And Bala is from Bugworks. Bugworks is developing a so-called NBTI, which is a novel bacterial topoisomerase inhibitor. This is a program that features a lead small molecule and a series of backups with a novel mechanism of action focused largely on the broad spectrum of activity targeting the escape pathogens. Bugworks is in the preclinical stage and heading towards a first-in-human program, hopefully next year. They've successfully conducted conversations with the FDA, and I'm looking forward to hearing what Bala is going to say about those interactions and how that helped them to shape their program. Next, we'll hear from Michael Bevilacqua from Amicrobe. Amicrobe is a company based in California. I should say Bala and Bugworks are based in India, and so we're covering quite the range of time today. Michael's company is focused on a novel protein that is topically or locally applied and focused on surgical settings, and initial emphasis on prosthetic joint infections. Next, we will hear from Matt Henn and Chris Ford from Ceres. Ceres, also in the preclinical stage, has an oral rationally designed fermented microbiome therapeutic, and they're focused on the prevention of antibacterial resistant infections, largely in transplant patients. Ceres, actually, like Bugworks and Amicrobe, is in the preclinical stage and focused also on a first-in-human program, hopefully next year. Then we'll transition to vaccines, and Javad Aman and his colleague, Catherine Bernard, from Integrated BioTherapeutics will talk about their very interesting 5-component toxoid vaccine for the prevention of skin and soft tissue infections caused by Staphylococcus aureus, both MSSA and MRSA. And then we'll hear from our colleague, David Steinmiller from HelixBind. HelixBind is a diagnostics company focused on direct from sample solutions. Here, I think we're going to hear from him today about a direct from blood product for assessing infections, including sepsis. Now I'd like to invite Bala to start his presentation. Thank you very much.
Thank you, Erin. Thank you, Astrid, and thank you, GARDP and CARB-X for this opportunity for Bugworks. I hope I'm audible. What I'm going to talk about is a very recent experience that Bugworks had in terms of having a pre-IND meeting and how that has been very educational, so to speak. So I've been told there will be a delay in the slide movement. I hope there it is. Okay, so I know how much the delay is. First and foremost, I would like to acknowledge a whole host of partners that we work with, without whom Bugworks doesn't exist. We work with multiple partners from the Bay Area all the way to Japan. And of course, tremendously grateful to our funding partners, CARB-X, and the government of India for a generous support, without which these programs actually would be very, very hard to move forward, especially for a startup out of Bangalore, India. So what does Bugworks, what does it do? In terms of a platform technology, what we started out with is called an elute platform, where we focus on dialing out the efflux-mediated resistance. So we start with compounds which have an efflux liability through a specific class of R&D pumps, do structural biology basically, and dial out the flux liability. And in the process of dialing out the flux liability, dial in the target mechanism. And in this case, what we're looking at is a potent gyrase inhibitor, which works both on the gyre as well as topo4 equipotently, and yet differently from the site of action of fluoroquinolones. So this is, broadly speaking, the platform of Bugworks. And what it covers is the exciting spectrum that we have, what Erin mentioned in terms of the focus, the critical, high, and the medium priority pathogens of the WHO and CDC list. And the Gyrox as a package and the lead compound actually is very potent across the entire spectrum, covering all of the resistance mechanisms. And that is the key excitement in terms of moving this compound forward. Where are we? Again, as Erin just mentioned, we're just in the transition point where we are close to selecting our first candidate for first time in human studies. And we've just completed the preclinical tox package, as well as the PK/PD studies, which allows us to look at in terms of what would be a possible therapeutic dose and then a therapeutic margin. And in that context, had a pre-IND meeting with the US FDA pretty much during the late stage of the preclinical development. And this meeting actually helped us crystallize a lot of things in terms of strengths, the weaknesses, the gaps, etc. And the context of today's presentation, the webinar, is actually some of the learning that came out of this pre-IND meeting. But before I go on to that, some of the things that is sort of a introspection. The decision to request this meeting actually led to a lot of learning as it is. What it did was actually organize ourselves, compilation of all the preclinical reports, QC them, identify gaps, and so it actually an enormous learning exercise just getting ready for such a meeting. And then start thinking of regulatory requirements rather than limiting to just exploring the science as the lead optimization progresses. This is often a trap that young drug developers like us, Bugworks, which is a first-time drug developer, which actually get trapped in terms of a lead optimization program. For example, the key example, it's something that an advisory board member cautioned us maybe a year ago: wake up to CMC aspects. And for us, the CMC is basically producing a bulk amount of the API. But as we started putting together in terms of the pre-IND dossier, we realized there are lots of things, and we learned along the way. And in a very lighthearted moment, I could even say some of us learned the difference between a drug substance and a drug product. So things like this is clearly a great wake-up call, rather than just knocking on the doors of an IND submission. Clinical development, thinking beyond the first time in human studies, laying the foundation for an investigator's brochure, and clearly laying the foundations for an IND package. So that's what actually the pre-IND meeting triggered a whole host of things that we could benefit. What did we apply? Just a nomenclature, the 505B1 application. Again, this is something of a learning for guys like me who are doing this. So why was it relevant? If you actually go to the website of the FDA.gov, you will actually see a variety of reasons. And interestingly enough, 4 of those are directly applicable. Clearly we're new to drug development. There are questions from us. There are pharmacological, clearly tox signals that we want to actually address and get a feedback. And it is a new molecular entity. So there are lots of contextual reasons why this pre-IND meeting became extremely important. So what gets submitted? Again, this is a learning curve, a steep learning curve, probably a laundry list for many people, but it's actually, I thought it's good to put in because it's something that we went through and learned on the job. A cover letter, a short synopsis, a list of questions, then a massive document, and then a slide deck actually for the pre-IND meeting. So multiple things actually you have to prepare yourself in terms of getting towards such a meeting. A broad timetable, again, this was a learning. It actually requires a good 2 to 3 months preparation. This is not something that we wake up and suddenly say 2 weeks from now we can get this organized. The actual timetable kicks when you submit the request letter, and then it's a 60-day process for where the pre-IND meeting actually happens. Within the first 10 days, you get an acknowledgement, and then within the 30 days, you submit the large briefing document. And then on day 55 or so, you get a feedback on that, and actually the presentation on the 60th day which actually happened for us on the 60th day, is a 60-minute meeting, and we only focused on the feedback that was received on day 55 and not to a whole host of the project presentation. And the minutes of the meeting does come within the 90 days. So that way, it's a very well organized, streamlined process in terms of the pre-IND meeting. So a couple of examples in terms of why this was such an educational thing. As I already alluded to, we became aware, got educated, actually started building the whole dossier for the regulatory aspects of CMC. And this was clearly a blind spot, I would say, for the ones who are within the discovery team. Probably a blind spot for many startup and first-time product developers, although quantitatively that blind spot may be different. So what was a clear learning was early engagement of CMC experts, particularly versed with the regulatory aspects as we move on, because you have to think much, much beyond the phase 1 studies rather than just limiting to crossing the threshold into the first time in man. The second example is a toxicology example. I mean, I'm giving you only one example. In fact, identified a key gap in one of the toxicology studies. So this was very useful because we had not yet completed the package, and so it led to a redesign of the study. And clearly, the feedback was a better risk management in terms of anticipating certain worst-case scenarios. And as product developers, we're always, in this realm of absolute optimism that everything will be hunky-dory. But the risk management and actually anticipating some worst-case scenario allowed us to better design. And in the end, when we completed the study, this was an extremely valuable suggestion. So these are just a couple of examples why a pre-IND meeting, even before you complete the package and get ready for the IND, is actually extremely useful for us. This is an interesting, for a majority of the questions, the feedback that we received was, further evaluation and determination of acceptability will be based on the information provided in the IND. And there were different perspectives. For us, not bad, at least no obvious red flags for the particular question that was raised. For someone else, it's like, only standard language. I wish we got a bit more feedback. The best was the investor, yay, FDA has not put a hold, let's go forward. So it's an interesting, different feedback, how it's viewed from different parties on the table. So my last slide, the conclusion is that this is extremely valuable in planning a drug development program, especially the questions are not fully answered, but the guidance is available on the website. These early interactions actually help prevent a clinical hold, which can arise downstream. With that, I hand it over back to Erin. Thank you.
Thank you very much, Bala. Great presentation, and love the levity that you inserted as well. Certainly a lot of lessons there. Okay, we're going to move to Michael now. Michael, please take it away.
Erin, thank you very much. Also, thank you, Astra, and everyone who helped to organize this conference and put it on in this challenging time. I'll begin by saying I agree with everything Bala just said. So we can, and moving forward from there, I'm going to talk a little bit about our experience at Amicrobe in building our clinical and regulatory strategy, which, just like what as Bala said, is still ahead of us, but it's trying to get our thoughts clear as we move forward. I think, as you will see, this is all about these molecules we call amicidins, and amicidins are synthetic proteins, synthetic amino acid polymers, actually ones that are made more like other polymers like nylon than like traditional biotech products. But you're going to see that they are, in multiple ways, they can be called non-traditional. Just waiting for, again, there. So I'll specifically talk about what we do. I'm going to try to point out 3 clinical questions that we found key in building our strategy, and then just a few statements on working with regulators. I've tried, and with our whole team, we've tried to make sure as we're doing each of these steps to think about what will bring us success, and success thinking beyond just getting into that first clinical trial or successful phase 1, what will bring long-term clinical success, regulatory success, and even commercial success as we build our strategy. So at Amicrobe, we are at this fun and interesting interface of material science and biotechnology. So again, our molecules are chemically synthesized, one-pot polymerization, but they come out as proteins. So we are designing and engineering proteins to prevent and treat infections all locally. The question is, why are we doing this? Well, I'm going to borrow here a statement from 1917 from a Nobel laureate Alexis Carrel and one of his surgical colleagues. And they were working in the battlefields of France with some of the worst infections in history, and said, infection at the outset is always local. And I think I would comment that if we look across the spectrum of infections, microbes really like local environments, especially wound environments. But today's antiseptics and antibiotics don't particularly like them. And remember that local infection that begins can progress. It can invade, it can go systemic, and you bring the patient to sepsis. And today, sepsis is still responsible for 1/5 of all the deaths worldwide. So we are trying to build the next generation of locally applied antimicrobials. Center to that strategy is the molecules we create have dual modes of action. It's inspired by innate defenses. As you think of your own innate defenses, whether you think of skin or mucous membranes, what you have is both a physical barrier or a cleansing process, as well as microbicidal activity. And we've designed in our molecules to try to achieve that dual mode of action. So, synthetic proteins, and we design them with a block architecture. Very simply, as you see, Amicidin alpha is the bigger of the molecules, targeting about 170 amino acids in length. Of the positive or cationic block, which is shown in the blue, and the red or hydrophobic block, as shown there. In the Amicidin alpha, it's alpha helical. It causes these molecules to self-assemble into fibrils, and they then form a hydrogel at very low concentrations or very viscous solutions. Amicidin beta, a little different. We designed this one to have greater surfactant properties, more for cleansing, forms micelles and stays in solution. The little table on the bottom shows that amicidins are pretty different, similar in some ways, but pretty different from traditional antibiotics or antiseptics, or even the previously tested antimicrobial peptides, which were all much smaller and not designed for physical properties. Based on the materials aspect of these molecules, with little or no excipient, sometimes just water, or you can add things if we want to change or again, adjust properties a little bit, we can easily turn amicidins into hydrogels or solutions, to dry fibers, thin films. We can also add other polymers to adjust properties, and even add other pharmaceutical agents for things like delivery or combined activities. So where are we targeting this? And this is, again, all about thinking in the future, thinking about what are our clinical trials going to look like, what is regulatory going to look like, and even what is commercialization, of course, going to look like. Well, we've targeted first surgery and trauma. One, we believe the need is now. There are a lot of infections. These infections can be quite serious. They can be life and limb threatening. There's also a tremendous number of procedures. There are over 300 million major surgical procedures annually. There's also tens upon tens of millions of traumatic events that open wounds. Add these things together, and it's pretty quick to get to over 1 million events a day that could be opportunities. And they're also, though, unfortunately for patients, there are opportunities for bad things to happen. And I'm sorry, this just backed up a little bit. Okay, so our lead investigational products are Amicidin alpha surgical gel, designed to be a barrier for local application to clean tissues, and Amicidin beta solution. This is the one that is supported by CARB-X for local application to now contaminated or already infected tissues. Alpha, dual mode of action, barrier and microbicide. So the physical barrier forms can coat tissues. Think again of a surgeon just beginning a clean surgery. They're cutting in, now that the underlying tissues are exposed, they get contaminated during surgical procedures, just from the air, from bugs that crawl, microbes that crawl in from the skin. And so the ability to put on a barrier that will also be a microbicide during the surgical procedure. Add them with that microbicidal activity, the dual MOA, we call it block and kill. Beta. Now it's when tissues are already contaminated. Think of major or even minor, but really we're thinking in this case mostly major traumatic events like open tibial fracture, or things that have gone further than that. Perhaps you've had a joint replaced. That joint has become infected. This is a very dangerous infection, like prosthetic joint infection. So now we need to do more than just protect. We need to actually cleanse and debride. It's designed to be a more aggressive molecule. So it's designed to help that surgeon debride, as well as break up biofilms, as well as kill microbes. Its dual MOA is cleanse and kill. All right, so what do we think about as we're trying to develop this clinical and regulatory strategy? They sound very basic questions, but they're not always so easy. In our case, what are our best clinical targets? We've had people recommend a lot of different things. Many people suggest go to very simple things, and some recommend more aggressive. We'll talk about that in just a moment. Which outcome measures will help with our determination of how well things are working? And very important to us, how much, how often, in terms are we applying our Amicidins during surgical procedures? Do we need them after surgical procedures? So, best clinical targets. Again, we've tried to get the smartest clinicians and clinical scientists we can to ask about this. And we've talked about a lot of different things that range across almost every single organ and tissue set, as well as different clinical practices. What's risen to the top are the things that are underlined. For alpha, mastectomy with reconstruction is a high priority for us. Also in the digestive system, open colorectal. For beta, we're really looking at hardware-related, orthopedic hardware-related infections, either after they've happened, like prosthetic joint infections, or even before they've happened, like open tibial fracture. What about the other outcome measures? And all I'm talking about here is beyond wound infection. Microbial counts are can be very critical to help us determine how well things are working. What about surrogate markers? But it's important to point out that things like being supported by CARB-X and work by others, these things are improving right now. The ability to do microbial counts in these settings is getting better. Okay, and so what about the other surrogate markers? Being able to put into a picture in order to determine how much, how often early in your clinical trials or even in preclinical work, so we can get a better sense of how much we need here. There's also interesting things in development, older things like composite scores like asepsis, newer work on analyses like door radar analyses, which help to look at clinical outcome measures other than simply wound infection. And the how much, how often, the third question. It's clinical research and development. The best we're going to have is an educated guess before phase 1. We need in our phase 1/2A studies to do great clinical work to help figure that out in advance of getting into our phase 2B and 3s. You all have probably heard the nightmares of things where companies get into their phase 3 and just didn't have it quite right, and that phase 3 fails and things go quite badly for them. It's really important to get this number right. In some cases, it may be straightforward. In our case, it's not all that straightforward, and I'm sure with many of your own programs, it's not that straightforward to determine how much and how often, but it must be done as scientists. And we also try not to forget, again, the commercial implications of what we do. Let's talk just a moment about working with regulators. I completely agree with Bala. I even go earlier, meet early and focus on key questions. For us, we had really surprising things happen with our consultants. So we brought in consultants that because of the way our molecules looked and what they were doing, we had differences of opinion on whether or not we were going to be regulated as devices or therapeutics. We had differences of opinion on whether we be regulated as drug or biologics. And of course, then the more standard things, what kind of microbiology, GLP tox. So we went quite early, and all I can tell you is that the FDA was of tremendous help to us. I also want to remember as we go forward that it's not just getting approved, especially for commercial outcomes. One has to consider the breadth of the indication. I was long ago a vice president of Amgen, but even before I got there, Neupogen was developed. This is an extraordinary product. It was launched in 1991 with, in today's dollars, over 300 million in revenue. 5 years was almost up to 2 billion. But look at its 2019 US sales. It's a pegylated derivative, still at almost $3 billion. What does it do? It prevents infection. It's a molecule designed to prevent infection in cancer patients. It had a great indication, and they worked it very, very well. All right, I'm going to close with just a simple comment or two. I'm a fan of the FDA. I'm a fan of the agency and the people there. They've done a great job with us. They also put on conferences and participate in other conferences. They offered us 2 pre-IND meetings, one for alpha and one for beta. Their feedback has been really, really helpful and thoughtful. So I think I would just leave you with that concept that the regulators can be a real help. And in that, I go even a statement even broader. Perhaps this is just because I've been around and doing these things for a long time, but I think as COVID-19 has demonstrated absolutely clearly, when it comes to preventing and treating life-threatening infections, all of us, the scientists, the clinicians, the drug developers, the regulators, we are really all in this together, and I hope we can all keep working together. I again thank the organizers here for putting this on, because that is how we will continue to work together. Thank you very much for the opportunity to present the Amicrobe story.
Thanks very much, Michael. And I can say with certitude that we were at CARB-X among those who were surprised at the FDA and some of their suggestions and partnership with you on beta. So it's a very, very interesting series of meetings. Okay, we're going to move over to prevention now, and I think both Matt, Henn, and Chris Ford are going to talk to us about series.
Yes, hi everybody. Thank you, Erin, for the introduction and the opportunity to speak, and CARB-X obviously for their continued support of our SEER 155 program, and of course, Astra, thanks to you to helping as well organize today's event and GARDP's efforts here. I'll lead off by talking a little bit about our technology and some of the key challenges we've had as we've moved from discovery all the way through to clinical development across our portfolio, and also speak a little bit about our SEER 155 program, and Chris will dig in to some of the details around how we've addressed some of the challenges that come with bringing a novel drug modality forward. So Seres is a company located in Cambridge, Mass. Our mission is to transform the lives of patients with revolutionary microbiome therapeutics. We are a clinical stage company. We have 2 clinical assets that are in late stage, one which just completed a phase 3, one which is in phase 2. And then we have other pipeline assets that are moving into the clinic, including our SEER 155 program that CARB-X supports in the very near future. We are a platform company. We've developed a method with which we can define microbiome therapeutics and move them into the clinic, and we'll talk a little bit more about that in just a second. So what are our drugs? You heard me say a novel drug modality, and what our modality is that we are developing consortia of bacteria that are designed to have specific pharmacological properties. So these are live biotherapeutics regulated by CBER. Our drugs are formulated for oral delivery using GMP manufacturing practices. Importantly, they're designed to target inflammatory and immunological disease pathways simultaneously, and importantly, we target multiple pathways simultaneously. So the drugs have multiple MOAs, and they're designed to establish colonization resistance to pathogens. The consortia of bacteria are not your typical strains that are used in things such as probiotics. Instead, what we've done is we've identified bacteria that are commensal in the gut and designed consortia that capture the full breadth of biology and functional diversity in the gut. And lastly, our drugs are actually designed to engraft. And by engraftment, we mean the bacteria in the drug actually populate the gut take up residence and become metabolically active. And this allows us to both restructure the microbiome as a whole and achieve durable impacts in the treatment. Slide's not advancing. Give me 1 second here. There we go. So, so our drugs are actually designed to rapidly restructure the microbiome to treat both infectious diseases and also modulate host immunity. So our drugs are orally formulated, as I pointed out before. And what we do is we identify disease targets and look for diseases where the microbiome in the gastrointestinal tract is susceptible to disease, and we identify how it's susceptible to disease. And then we design consortia of bacteria that will restructure that microbiome and then lead to the production of microbial-mediated metabolites to engage the host and modulate host immunity or to compete against specific pathogens. So the first step in that is the engraftment of the bacteria, which is that population of them in the gut. We've defined that as a measure of pharmacokinetics for this novel drug class. And then measures of pharmacodynamics include restructuring the microbiome composition and then, of course, the change in that function. And importantly, our consortia are designed to target multiple disease-relevant pathways simultaneously. Our view as a company is that one of the limitations of many drug modalities out there is that they target single pathways for diseases that are multifactorial. And so we specifically design our drugs to be able to modulate multiple disease-relevant pathways simultaneously to achieve greater efficacy. So we have a broad pipeline, which includes efforts in infectious disease, inflammatory disease, and oncology. As I noted before, we have a phase 3 asset, our SER-109, which is for recurrent C. difficile infection. We're super excited about our SER-155 program, which is the program that we're working on in collaboration with Memorial Sloan Kettering and is supported by CARB-X. And in this case, we're looking at infection, the prevention of infection and bacteremia in subjects who undergo hematic stem cell transplant. And we've designed a composition that can prevent infection of certain antibiotic-resistant bacteria, and that can also modulate host immunity to have greater receptivity to the stem cell grafts. We also have a program in ulcerative colitis, which is in a phase 2 study. We have another UC program, which is about to enter the clinic very shortly in Australia and New Zealand. And then we have an oncology program as well, where we're currently running a phase 1b in metastatic melanoma in collaboration with MD Anderson and the Parker Institute of Immunotherapies. So we have established proof of concept for this novel microbiome therapeutic approach. As I said, we've recently read out our SER-109 phase 3 study for recurrent C. difficile infection. The results were incredibly encouraging. Top line efficacy, we saw a highly significant 30.2% absolute reduction in the rate of C. diff recurrence. This was in multiply recurrent C. difficile patients. That translates into a sustained clinical response rate of 88.9% on drug versus 58.7% on placebo. And for those of you who are familiar with drug development, our number needed to treat is approximately 3, which is outstanding for a phase 3 asset. Importantly, there were no serious drug-related adverse events, and the overall incidence of adverse events through our top line endpoint were actually similar between the 2 different arms. We have 2 publications on the phase 1 and phase 2 data if you're interested in seeing that. We also, in the context of ulcerative colitis, have read out our phase 1b study, and there we saw a significant 40% clinical remission in arms with a preconditioning and daily dosing. And as well, we saw a strong safety profile, and results from this study were recently published in Gastroenterology. So as we think about the core challenges to developing a novel microbiome therapeutic and things to think about, my other colleagues on this panel have already touched on numerous points that I couldn't agree with more. And we have certainly experienced in our course of drug development at Seres. But the first challenge is that, especially when you're focusing on an emergent science, it requires heavy investment in core technologies and translation. Something we learned at Seres was that there were not existing CROs and vendors and other things for much of what we wanted to do. And so we had to seek investment to be able to develop those various different tools. And one of our key solutions there was to actually de-risk discovery by leveraging a reverse translational strategy. And what does that mean? It means that we actually focused on studying the microbiome in human subjects and then leveraging non-clinical and preclinical data to support the findings that we are observing. And what that does is it de-risks the translatability of the drug modality. But unfortunately, that does require significant investment. And there are various different tools that we'll talk a little bit more about, and Chris will hit on, that were used to be able to enable that. The second challenge is really a lack of commercial vendors for manufacturing and quality activities. I mean, you've heard several of my colleagues on this panel talk about the importance of CMC, and I can't emphasize the need to think about manufacturing very, very early. In the context of microbiome therapeutics, it's very comparable. I like to think about it as comparable to gene therapy. Nobody thought about manufacturing until they had to think about manufacturing, right? And the companies that succeeded are the ones that had invested heavily in manufacturing. And so what Sirius has done is we've actually invested since inception of the company back in 2012 in manufacturing. Actually, the three longest tenured people at the company, original hires, was actually myself, who was building the platform, our head of manufacturing, and believe it or not, the third hire at the company was actually a regulatory person, even though we were years from the clinic, because we wanted to start thinking about what the regulatory landscape might look like. And so, you know, our solution there was to think about this early and invest in that. And thankfully, we had investors who were strongly supportive of that concept. The last point I'll make is really establishing regulatory expectations, particularly when you're pursuing a novel product. And again, you've heard folks say, engage early, engage often. And I couldn't emphasize that more. We engage regulators very, very early and took pre-IND meetings. And in addition to additional strategies that we deployed as we've developed this new drug class. First is think about your patient populations, because something that Sirius has leveraged is getting orphan drug designations or fast track designations for our drugs. And we achieve that by thinking about how we think about the patient population we're targeting and where the greatest need is. And what that allows you to do is have much more frequent engagement and responses from the agency. The other thing that we did is Sirius, in collaboration with a couple other companies in the space, established the Microbiome Therapeutic Innovation Group. And that organization is pre-competitive, where companies working in the space, we've come together to work collaboratively and engage the agency collaboratively to define, in a sense, the rules of the game for this new drug modality. And that's been very seen very favorable by the agency. Just last week we had a meeting with Peter Marks and all his lieutenants where we discussed some of the key challenges and just got feedback and dialogue with the FDA about, you know, kind of these challenges that we're facing and understanding how they're thinking about it. So with that, I'll turn it off to Chris to speak specifically about some of these additional challenges.
Great, thanks, Matt. So as Matt highlighted, really it requires investment across the pipeline here to enable this kind of work. Sorry, going back one slide. And really we start from disease target and identification and build out tools across our pipeline to enable really a concept, reverse translation concept, and translating that concept all the way through to regulatory and clinic. On the subsequent slides, I'm going to just hop into a couple of these specific topics, but I'll provide a broad overview here, whereas as Matt has mentioned, we start with this reverse translation concept. And really for us, what that means is that we need a clinical sample biorepository to understand how the microbiome is interacting with any given disease and maybe playing a causal role. And so having a broad and deep repository there that we maintain at high quality has been central to our early discovery process. The next step in that process is, of course, understanding what it is that we're seeing in those samples. And we started there with publicly available tools and quickly realized that for us to achieve the kind of resolution and throughput and sensitivity that we needed to build out proprietary tools in that space. And so that's one that I'll highlight on a subsequent slide. And that work is done not in isolation, but with support from world-class collaborators. And I think we've seen that theme throughout today's webinar, is that building out a support team outside of your company is central to your success, right? And certainly one way in which we've been able to do that is through our interactions with CARB-X, but we have a collaboration network that extends beyond that, and I think has enabled development across each of these categories. Additionally, to then translate our concepts into reality, we need a strain library that allows us to enact those designs and then subsequently test them. So we compile compositions of bacteria that we think may have therapeutic impact, and then evaluate those both in vitro, ex vivo, and in vivo disease models. And we use the interaction of those 3 model systems to understand how any given composition of bacteria may be able to have a clinical impact. I can't underscore enough Matt's points about CMC. Being able to translate, and we've only got that represented here by 2 small circles, like a flask to grow up our bacteria, and then they're in a pill. And that is a vast oversimplification of the amount of work that goes into translating these drugs from concept and R&D into reality that's ready to hop into clinic. And that investment in CMC has paid off not only in our ability to really have access to a broad set of our strain library where there's a lot of unknown biology represented there and has required a lot of discovery, but also then to take those compositions or those products to the FDA or to other regulatory agencies, enable them to understand that we're putting forward a quality GMP level product. And that's been the underpinning of our interactions with regulatory agencies around the world. We started with the FDA, and to Matt's point, leveraged interactions or pathways like orphan drug designation, and once clinical data was in hand, fast track and breakthrough therapies to enable this like early and often approach that we've heard about so much today already. And then our early phase studies are really focused on establishing safety and PK while working to hone our clinical measures of efficacy to enable those late stage clinical results. And Matt has already highlighted our recent SER-109 phase 3 result that really was the result of extensive clinical development and adaptation across that clinical development path. So I'll hop into some details here and just start by saying that the microbiome, especially 10 years ago when we founded Ceres, was really this black box, right? People didn't know how you might translate a complex ecosystem into a therapeutic. And so we started with publicly available databases to enable those analytics, and then quickly realized that comparing those publicly available data sets to our own strain library and our own data sets revealed there are a lot of gaps. And I think that's really well illustrated by this phylogenetic tree we have here on the right. So phylogenetic trees are sort of a visual way of representing bacterial diversity, and things that are further apart on the tree are more distantly related to each other. Here we've zoomed in on one particular group of bacteria, and in public data sets, this area of the phylogenetic tree is represented by only 3 species, and we've got those 3 named here. That's what you're seeing in text. As we compare that back to our strain library, shown here with blue stars, or to genomic signal, where we have computationally assembled genomes of things not yet cultured, which is what we have in both green and pink squares, we see that there is a wealth of what some people call bacterial dark matter, represented by this otherwise simple portion of the tree with only 3 species. So being able to dive into these areas of unknown biology and use those to help drive our early stage signals has been really valuable, and that investment continues to pay off. One way in which that has been acutely true is in using not only the strain library to work backwards to understand where our gaps are, but to use those analytics to work forward and build out a strain library. And so now you're seeing a much more full version of a phylogenetic tree. This covers the set of gut bacteria, or the set of bacteria found in the human gut. And the rings around this tree, the first ring just shows you the taxonomic groups. And I know it's a little bit hard to your screen. You might have to lean in some, apologies. But the outer rings then show you where in our species collection or in our strain library we have at least a single isolate shown on this first track, or multiple isolates shown here in orange. And I think building out this proprietary wholly owned strain library as a resource is really, it serves as our what other companies might use as a chemical library. This is the primary engine by which we then enable discovery and translation. There is a significant amount of investment that goes into just being able to anaerobically grow and culture bacterial species from this broad swath of diversity, right? This is required not only importing tools from academia and known culturing techniques to access some of these harder to reach regions of the tree, but also building out new techniques and new technologies to enable this diverse accumulation of the library. And I think one way in which that's well represented is by looking at the set of species that are present in our strain library that are actually on the NIH's, the National Institute of Health's species list of most wanted species. So after an effort was undertaken to profile the microbiome of healthy subjects, they realized that in that metagenomic data, there were nearly 500 species that were represented in human guts that we just didn't have strain isolates for. And in our proprietary collection now, we have built out greater than 60% of those, and that number actually could probably be updated today and be a little bit north of that, because this is an evolving space in which we're constantly driving and investing. The next spot, and again, I summarized this very succinctly with two little circles up here, but really our ability to build out bioprocess, which is this concept of taking a strain in the library and enabling it to be able to grow at titer with the phenotypes that we want it to in a way that is GMP compatible is no small feat. It's required investment in high throughput formulation screening, so to identify GMP compatible conditions under which these strains can be grown, while also optimizing the yields from each of those conditions to enable a product that can then be expanded and serve as the primary drug substance by which we'll deliver our drug product. And then in addition to investing in bioprocess development, it's also necessary to invest heavily in manufacture and in quality, right? So the quality by which you deliver your product and ensure that what you're delivering to patients is easy to overlook and underestimate as just a box checking exercise, but really understanding that what you're producing and delivering is what you think it is, has just incredible value, both in reassuring clinical sites that you're delivering a product to them that is safe for their patients to do these first in human studies, to reassuring and driving conversations with the FDA to do the same, and to giving yourself the confidence that your product being tested is what you are hypothesizing it to be. And that kind of clarity and investment has really, I think, delved from a partnership with both consultants, but particularly with the FDA, as we're doing some initial technology development here in this relatively new space, talking early with the FDA and often to help understand what they expect out of these kinds of products and assays has been incredibly valuable. And so we started those conversations with the FDA and have since expanded them to take a more global approach. And now we've had similar conversations with Health Canada, with EMA, as well as member states in the EMA. I think it's easy to underestimate that the impact of those member state opinions in going into Europe more broadly. And then more recently, we've also started conversations with Australia as well as New Zealand through interactions with the TGA and MedSafe. And I think really those interactions have focused on defining their expectations and making sure that they understand where they don't have yet expectations because of the novelty of the product, proposing solutions for potential challenges that we're going to be facing, right? So going in with not only questions, but proposed solutions has been very valuable and a great way to start that conversation. It allows us to determine not only the requirements for non-clinical safety and tox, especially when you're working in new products, but then also to establish that early approach to clinical safety. And that then nicely has dovetailed into the ability to define metrics and expectations for PK and PD in an otherwise undefined space, right? So as Matt highlighted on one of our earlier slides, we think of pharmacokinetics in this space as being the engraftment of our bacteria into the subject's GI. And pharmacodynamics as being the impact that engraftment has both on the broader microbiome, but also on the production of key metabolites, which may be driving biological activity. And I think until we started using that language, that wasn't really a defined concept. And so putting that forward and using known terms, but applying them to your space has really translatable value for helping to drive conversations with regulators. I'll stop there because I know Astrid's trying to keep us all on time. But I do want to just finish by thanking the many patients that really have enabled all of us to do the work that we're doing, as well as the clinical sites that enable this kind of in-human research. Of course, the team at Ceres, as you guys are seeing, this is a broad effort that requires a lot of investment across the board. And then, of course, our funding agencies that have brought us together today, including CARB-X, who's been a really valuable partner, particularly on our SEER 155 program as we make that transition in real time right now from preclinical to clinical. So with that, I'll hand it back over.
Thanks a lot, Matt and Chris. Definitely, you guys are pathfinders in this area. We're learning a lot from you. So now we're going to transition to vaccines, and I'm going to hand it over to Javad and Catherine.
Well, this is Javad Aman. I'm the President and Chief Scientific Officer of Integrated BioTherapeutics, a company in Maryland, and we're fortunate to have CARB-X support for development of a Staph aureus vaccine. What I would like, during this, first of all, I would like to thank the GARDP and CARB-X and the organizers for this opportunity, and also I have to thank the previous speakers because it made it a lot easier, and I don't need to repeat a lot of good advices that they gave, particularly around early engagement of the regulatory agency. So I would like to focus primarily on some of the unique challenges of being of developing a vaccine, and especially a vaccine for a very, very difficult pathogen. Just a little background on our company. So we are a boutique biotech, and the core of our business is to discover and take product candidates into phase 1 and hopefully partner with the pharma. And it's primarily based on a rational design. We work both in the vaccine and monoclonal antibody therapeutic space. And we have a portfolio that is focused on a number of emerging infectious diseases. We have lab and office space in Vivarium here in Rockville, Maryland. One of the things that we did very early on was that we focused a lot on non-dilutive funding. We have raised over the past 13 years about $85 million from NIH, DoD, and then more recently, since 2017, from CARB-X, and then we raised our first private equity funding from Novo Holdings Repair Impact Fund. This business model focused a lot on getting a government support, NIH support, has allowed us to really spend a lot of time on the science and develop our product concepts, without the pressure of the investors, for rapid results. So as I told you, we wanted to focus a little bit on, what are the very unique challenges of developing a vaccine, especially for a disease like Staph aureus, or a pathogen like Staph aureus. As you know, Staph aureus lives with us for a very long time, and all of us come across this pathogen in one way or another. It causes a variety of diseases. Each of these diseases are really unique in their own way. So when you are developing a vaccine for a pathogen like this, the best approach that we thought we have to follow is a reverse engineering of the development plan. Let's look at the end of the journey, and that is providing a solution for the patient. So who are your patients? Staph can cause a lot of different syndromes, and this could be what we hear most about, especially for MRSA, are these surgical site infections, bacteremia, endocarditis, biofilm, a very big problem that causes the prosthetic joint infections, the catheter infection, implant infections, breast implant, and so on, and also these community acquired MRSAs, pneumonias, or osteomyelitis, a variety of diseases. Each disease is different. And then look at the your competitor prior experience. We looked at what has been done in this space? Everybody went against these, high value targets. And for example, Nabi did their clinical trial in end-stage renal disease patients, and they failed in phase 3. Merck went against surgical site infection in elective cardiothoracic surgeries. They failed in phase 3 after vaccinating 8,000 people. Pfizer went after this, a specific prosthetic joint infection in spine surgery with a prosthetic device, and they failed after vaccinating a few thousand people. So this by itself just shows the challenge of developing a vaccine as opposed to a small molecule. Small molecules a lot of times fail in phase 1, phase 2 before you spend tens or hundreds of millions of dollars in a phase 3. Vaccines are often safe in phase 1, phase 2. They are immunogenic, and they don't fail there typically. But then phase 3s can cost like 500 million, 400 million before you even know that they don't work. So it's very important to think about that end stage. So, and think about a disease that has sufficient attack rate that you can actually study, and it's manageable without an enormous expense. Now, so when we look at the entire space of Staph aureus, or the spectrum of Staph aureus diseases, you see that 90% of them are actually skin infections. And most of these skin infections are they resolve by themselves, or after a incision and drainage. There are up to 16 million visits to emergency departments or doctors' offices. But there is a big problem there, and that is that many of them cause recurrence. And then we took a deep dive into the history of Staph aureus, and we found even clinical trials in the back in 1929 that help us actually devise our strategy. So to make it short is that we decided to go after recurrence of skin infections, which has a very high attack rate. And then the question is here, what is the disease exactly? How do we call it? We look into the literature and FDA history. We see that okay, there is an ABSSI, but that disease was invented actually for antibiotics. So we had to actually invent a new disease, call it recurrent Staph aureus acute skin infections. And so we built that concept and went to FDA with that concept, and then work back from there. So what is our clinical strategy, and now what do we need to do to get there? And we had our pre-IND meeting very early, and Catherine, my colleague, will speak about that shortly. So an important, sorry, I have to go back. An important question here was what do we target? Because Staph aureus has a lot of virulence factors, and all the previous failed attempts have targeted the surface of the bacteria. So we, again, driven by clinical and epidemiological findings that have been published over the years, we came to the conclusion that the best way to prevent recurrence of skin infections is to target all this variety of toxins that Staph aureus produces and basically creates a smoke screen, and causing the immune response not being able, or immune system not being able to mount a protective response. So from there we went back to actually designing the immunogens. So sorry, this delay actually causes a little bit of time. So we designed these 5 proteins that are toxoids that target 7 different toxins of Staph aureus. By cross protection or cross neutralization, they cover up to 15 toxins. And we came up, as I said, with this clinical indication, prevention of recurrent acute skin infection. And I have to say, actually, in the process of refining this clinical indication, we were enormously helped by CARB-X advisory boards. We received this CARB-X grant. In the base, we completed the proof of concept studies. It was a very short period of base, 6 months. And in option 1, we completed all the preclinical efficacy package, bioanalytical, manufacturing, process development, and also we held our pre-IND at that stage, and that was extremely helpful. The advice that we got, and we actually did not limit that to FDA. We went with the help of CARB-X. We went to Germany, to PEI. We went to UK, MHRA, and those advices that we got from there was enormously helpful to further plan the option 2 and option 3, which is focused on manufacturing, safety, toxicology, and we'll end with IND submission. We are now in the middle of this option right now. And our phase, we also worked through not only phase 1 clinical trial design, we also designed our phase 1, not only for phase 1, we also designed our phase 2, which will be actually an efficacy study in a limited number of patients. And these patients are those who come to the clinic and they have a high risk of getting recurrence. So with that, I'll pass it on to Catherine. Catherine, could you please take it from here?
Thanks, Javad. So as all the speaker has identified, it's very important to talk to the FDA. And I would say sooner rather than later. You don't want to have your program development too far advanced and then got feedback that you didn't do the appropriate thing. So with the Staph aureus vaccine, we decided to go to the FDA before the GLP talks because, as Javad mentioned, vaccine are special from a preclinical standpoint. You don't have the whole the normal GLP talks. And so we wanted to make sure that from an efficacy standpoint on the preclinical study that we had conducted was enough to support the talks. And then the design of the talks was sufficient to support the phase 1 study in healthy volunteer. So we went to the FDA and we had a very good interaction, very discussion, open scientific discussion on what we needed to do and the design. And we reached agreement on the design of the study. Part of the discussion also that we start engaging the FDA was to make sure that they recognize that the acute skin infection, which is the indication we are looking for, was kind of an unmet medical need because, and I'm sure you all know, once you get an indication or at least a recognition from an FDA that you get an unmet medical need, it kind of opens doors for a breakthrough, fast track, and all of those accelerated programs that are available. So we also get the agreement that the product as well as the indication were unmet medical need. And part of the pre-IND, we had also a synopsis because when you go to the FDA very early, sometimes it's difficult for them to let you know that your tox is okay if they don't have anything from a clinical standpoint. They cannot say, yes, it's okay, but then you're going to do, I don't know, a 2-year study versus, and you have only a limited number of talks to support your program. So we reach an agreement. And something that is also very critical is when you receive your pre-IND or your FDA meeting minutes, they are binding. So basically, you could, go back to them and said, we have discussed, and this is what we have reached an agreement. If no other data are available, they'll okay with that. The problem we had is we wanted to go further, as not just have an agreement on the phase 1, but we wanted to make sure that also from the phase 2 and 3 we were on the proper track. And so in order to do that, we went to the European regulators. And Javad, if you could move to the next slide, please. Thank you. So we use the relationship of CARB-X with the German Center for Infection Research to help us setting up a meeting with the Paul Ulrich Institute in Germany. And so we went there this year. And in parallel, since we were in Europe, we had also a meeting with the MHRA, so the UK agency. And pretty much we had the same type of question up to the phase 1 study, and then we had a propose for a phase 2 and phase 3 study, and we wanted to reach there. So with the European agency, you never get, you don't get an agreement in the sense that their meeting minutes are not binding. So they could give you any advice at the time. It may not be valid within 2 years by now, but at least we get some feedback as, do we need 1 phase 2, 2 phase 3, 3 phase 3? So, the ballpark of the clinical development. And again, both of those meetings were very, very helpful and discussion on the science as well as the regulation. They did agree as well on the GLP tox design. So we are good to go with 1 study across at least Europe and US. And then they give us some feedback on, what they would like to see from a phase 3 study, from a safety database, like 3,000 patients or more. So all of that was very helpful. And we also get the agreement that the acute skin infection was also an unmet medical need. So overall, my advice would be, yes, definitely goes to the FDA for a very specific topic. So GLP talks, first in men and things like that. But if you want to have a broader view of your clinical program, it will be more difficult early in the development with the FDA than it is with Europe. And so that's our message to go home. And I'll let Javad take the conclusion, please.
Thank you very much, Catherine. I thought basically this is the conclusion of our presentation, and just wanted to acknowledge all the people who have been involved on our end in our lab, our advisors such as Bruce Forrest, David Talon, Jean Lee, Lloyd Miller, Vance Fowler, and Richard Proctor. They were really some of the leaders in the field. And of course, this would not have been possible without the support of CARB-X and NIH. Thank you.
Thank you, Javad and Catherine. Really interesting talk. Okay, and we're going to end the presentation part of this webinar in diagnostics. So David, please start your presentation.
Thank you, Erin. Thank you, Astrid. Also, thank you, CARB-X and GARDP for organizing this webinar. My name is David Steinmiller. I'm the Chief Business Officer of HelixBind. We are an in vitro diagnostics company located just outside of Boston, in the United States, and we're supported in part by CARB-X and the NIH. Trying to get the, there we go. So I'm going to talk a little bit about our rapid molecular diagnostic platform for the direct from specimen characterization of invasive infections. The first product for Rapid is Rapid BSI. It's a direct from blood test that identifies the most common pathogens associated with sepsis, both bacterial and fungal at the same time, provides single CFUs per ml sensitivity across the panel, and again, it's direct from blood in less than 3 hours. And a rapid test like Rapid BSI is quite easy to use. You basically take an EDTA tube from the patient, snap it into our cassette, which is shown there in the picture, slide into our box, and the box does all the rest. The system was designed for use in pretty much any setting, and Rapid BSI is really the first of quite a long pipeline of products we're working on, both for bloodstream infections and also for other matrices. I want to talk this presentations about our interaction with the FDA, which has been quite productive. This year we were awarded or designated a breakthrough device, but we've been speaking to them for a couple of years. And a crucial part of that is our pre-sub meeting, which is the details of this talk. And I want you guys to think about the pre-sub very much like you would think about the pre-IDE for therapeutics. The regulatory pathway for a diagnostic, it's considered a medical device. Medical devices are classified by complexity and risk from class 1 to class 3. Most diagnostics, including ours, are class 2. And as a class 2 device, a big part of our interaction with the FDA is determine the pathway, whether we're a 510K, which is probably the shortest pathway for a diagnostic, or a de novo or a PMA being the longest. And it's in the developer's best interest to obviously make that as simple as possible. Shown there is a list of some of the documents that we're going to provide in our final submission for clearance. And all of these items are open for discussion as part of the pre-sub meeting with the FDA. And I'm going to really focus here on the analytical performance testing and the clinical performance testing, which we had to establish with the FDA to support our indications for use. To start out with our analytical studies plan, this might look like preclinical studies for the therapeutics guys. This is quite a large body of work. Each of these studies can be extremely complex and, if not designed well, very burdensome. So I would actually say a majority of our interaction with the FDA during the process was on very tightly defining each of these studies. And just to give you a sense, our analytical package is probably 5 times the size of what our clinical package is. And what's our clinical package? So our clinical study is 1,500 patients or more, suspected of sepsis and consented. And the sample that we're going to test on Rapid BSI will be taken from the exact same stick as blood culture, and that blood culture will be our comparator method. Now, for our clinical study, since we're looking at all patients suspected of sepsis, because that's on our label, unfortunately, or probably fortunately, most people suspected of sepsis don't in fact have a bloodstream infection, which means that this specific study is actually quite good to demonstrate the specificity of Rapid BSI. It may not be sufficient for sensitivity. You can imagine across our panel of 21 pathogens, many of them or some of them may not appear in any substantial numbers in our clinical study just because of rarity. And so therefore we have added an additional arm where we provide the clinical sites spike samples across the clinical range for each of our pathogens. And again, it's 21 panel pathogens. And we also provide some samples at or below our LOD, our level of detection. So now I'm going to talk about some of the challenges and opportunities that arise from this clinical plan and our analytical plan. The first of which, and there are many, is how in fact do we do this spiking that we need to do to provide the sites these samples at or near the LOD. This is quite difficult. One key aspect of Rapid is it only detects intact microbial cells. Now this is in contrast to many molecular diagnostic assays that detect any DNA in the sample. Our choice to go after intact cells only is by design. Detecting free-floating DNA will open you up to false positives, which is common among molecular diagnostics. Unfortunately, because we only detect intact cells, we cannot use frozen samples, and that's because some bacteria do not necessarily survive the freeze-thaw process. So we have to figure out a way to provide our sites non-frozen samples in a timely manner at basically a handful of cells in an ml of blood or in 10 ml of blood that they can test. And the technical challenges for this are probably too large for me to go into. So we sat with the FDA and had quite a long discussion about how to do this. What we were going to do and what the FDA agreed to was to only use local sites so that we can prepare the samples at HelixBind, deliver them same day for testing. And we spent a lot of time on exactly the conditions of storage during that short time period. The FDA really wanted us to do in-depth testing of what the load looks like in those samples during that time period, both to protect from cell death, but also to ensure that there's not growth, which is a way that I think a developer could probably queer this study if they wish to. So to determine and ensure that the samples remain as they should be during the study period. Very happy with how that turned out with the FDA. Another challenge, and perhaps a bigger challenge, is with our comparator method, culture. Now, culture is known to miss cases of bloodstream infections in certain instances, and some examples are shown here. These are from our clinical studies. On top are patients who were on previous antimicrobials. On the left, you can see the results of rapid. That's our entire. That's a panel. Each of the bars on that panel is a signal from a different probe. And you can see these patients were positive for Staph aureus on rapid. Both these patients were culture negative. If you look at the middle chart, you can see that the signal on rapid was well outside the normal range and certainly well outside what could be a contamination. After the review of the patient charts, it was identified that both these patients were on antibiotics, anti-staphylococcal antibiotics, in fact, and both had previous positive cultures. So it's not proven, but it's quite likely that Staph aureus was still in the blood of these patients at the time of testing, and that it's possible that the culture, the concomitant culture, was interfered with. Both these patients had very poor outcomes associated with these infections. Below, I show some graphs about polymicrobial infections, which are also problematic for culture. On the left, you can see a polymicrobial that culture caught and rapid caught as well. And they were concordant. And on the right, you can see an instance where culture detected a single organism, but rapid detected a polymicrobial. In both these cases, with the rapid cassette, we can capture all the material from the assay and analyze it afterwards. And we did that. We do that with all of our positives, in fact, during studies. And by bidirectional sequencing, we found that the rapid results in both these cases were confirmed. So that's kind of what we're seeing in studies. For our clinical study with the FDA, we know we're going to see things like this. And it's incumbent upon us as a developer to identify when culture is presenting a false negative and when perhaps our assay is presenting a false positive. And to be clear, as I mentioned before, molecular diagnostics are known to provide false positives, particularly if they are not focused on intact cells and are looking at free-floating DNA that might represent a previous infection. So we needed to work with the FDA to address the fact that we are going to see what we consider to be false negatives from culture and demonstrate that our method is correct and those are false negatives. Unfortunately, there is no referee that we can go to, no other technology that can accurately measure or detect direct from blood infections, particularly at the low loads that we're looking at. And so what we put together is a process to analyze the discrepant results. A key aspect of this is that not only do we need to use this process for discrepant results, but also for some number of our concordant results as well. Statistically, that is important for this to be a valid analysis. But we're going to use an external review panel, and they're going to look at patient records, and they're going to look at the results of bidirectional sequencing. And I think we put a lot of time with the FDA, had some productive back and forth on exactly what that is going to look like. So again, we're really happy with this. We see this as a big risk for our clinical study, and given that analysis of discrepant results, we think that's going to reduce that risk quite a bit. Another substantial challenge for us are external controls. We need to provide external controls to all of our sites for testing every day of our clinical studies, but also all of our customers to test proficiency, ensure that the instrument's working. Every new lot of cassettes we provide to customers, they need to test with external controls. And the external controls basically represent what a sample would look like, but they're in some way, it's something that they can run whenever they want. The challenge here is very similar to the challenge that I described for providing spike samples to clinical sites. How do we provide something that they can reliably recover at the loads that we detect, the low loads we detect? It's certainly a significant challenge, and it's actually quite more difficult for external controls because these need to be something that the sites can store on site and use whenever they want. And so this is ended up being a huge investment internally on how we were going to do this. The strategy we presented to the FDA was to create an artificial matrix that's like blood, along with a technique to stabilize microbial cells, and crucially, an air-free, disposable and process where the hospitals can reconstitute these and use them without introducing contamination. And again, but cons are on our panel, so you can imagine how easy it would be to create or introduce contamination if you're not careful. The FDA was amenable to our strategy, gave us a lot of great feedback. This is an ongoing discussion. We've had one pre-sub with the FDA. We will have more. One of them will be about the external controls as we determined exactly the loads and exactly the breadth that we're going to cover on our panel with these controls. And finally, this is my last slide, but I'd say the most important part of our interaction with the FDA, starting from our first informational meeting, was getting them acquainted with a new technology. And RAPID incorporates a plethora of novel technologies to do what it does. And getting these guys on board is a process. They've never seen this stuff before. Fortunately, the FDA is staffed by scientists who are just as geeky as we are, and so they were certainly willing to listen and I would say enthusiastic to learn more about what we were doing. An example of one of the many ways our technique differs from standard assays is our detection strategy. Rather than detect single-stranded DNA using something like hybridization, we detect double-stranded DNA by an invasion process utilizing artificial nucleic acids. And this invasion process provides quite a few advantages. One of them is specificity. If our probe is off our target region by a single base, so there's a single base mismatch, because we're invading double-stranded DNA, the competitor strand is right there as part of the DNA, it outcompetes us, so it gets popped out. And so if a single base will mean we do not bind, and that single base specificity, that's an exquisite amount of analytical specificity, which is really unheard of for most DNA detection. That allows us to do a lot of things that standard techniques can't do. And one example of that is thinking about balancing inclusivity of all the strains within a species you might be going after with the exclusivity and not detecting other species. And that's something that every molecular diagnostic test has problems with, and we frankly don't. And so given the fact that this is a new technology and with new capabilities, we spent a lot of time preparing and discussing with the FDA these capabilities, provided a lot of in silico and in vitro data. And we feel the result of that is that our studies, even though it's a new technology, are not substantially more than one might expect for a standard technology. There's some more things we have to do, but nothing that was overly burdensome. And we also feel that as the RAPID panels expand dramatically, laying the groundwork for this up front may reduce the burden of some of our future studies. So again, we've had phenomenal feedback from the FDA. We really like the interaction. I'm just going to echo what everyone else has said: is go early if you can. They are extremely helpful. And finally, I want to thank CARB-X and GARDP again for preparing this webinar.
Thank you, David. Really interesting. I think most of us really have no idea what is involved with regulators and diagnostics. So really, thank you. Great talk. Okay, now we're going to do a little Q&A, and for those of you, online, you can submit them through the chat function. I'm going to ask the speakers to turn on your webcams. Everybody can see you. Okay, great. So in no particular order, we had a question come in that was directed to Bala and Michael, but I actually think it can be directed to all of you, which is, have you got a strategy for fail early, and what does that look like?
Good question. Well, the whole talks package is actually looking at it from a fail early, right? Because you're looking at what kind of clinical risks that one can anticipate and then plan the preclinical studies based on that. Anybody else, please jump in.
I'm happy to add something there, Michael, if you want to speak. Go ahead.
No, I was just I agree with Bala's comment. It's always, of course, an issue to look, and I just want to put the complementary statement of that I mentioned, how much, how often. When you're dealing with novel products and you're going into novel settings, it's not always clear. Like for us, how much of our product has to be applied, how often it has to be applied, and what that will be. And we are always looking at the very old and standard issues of effectiveness or efficacy and toxicity. So I would say that the thing I am most concerned about that in that regard is picking our clinical setting, and then the how much, how often. And I'm sorry, but I'll just give one little personal experience. Long ago, when I became a vice president of Amgen, we took over a company called Synergen. And they had a product that I knew quite a bit about. It was an IL-1RA, which was blocking IL-1. And we took it over because they failed in a study of sepsis. And their company largely collapsed. And that product succeeded in a study of rheumatoid arthritis after we took it over. So again, this issue of failure and success, it's one to be thought about really carefully, because failure in one setting doesn't mean your product is bad. It may mean either you chose the wrong setting or you didn't use the right amount. So just a side comment.
Yeah, let me if I can, I'll add a little bit on top of that, which is, I think it's important in drug development to de-risk your process. And I think what I would encourage everyone to think about is how you de-risk your technology, or how you de-risk really just moving forward. And I think it's about building in important milestones along the way, and being true to yourself and defining what you need to see to declare a success, but at the same time making sure you're always looking closely at the data. I think a perfect example is Seres itself, right? I mean, our lead asset, our phase 3 SER-109 program, which just had a blockbuster phase 3 readout, right? That drug failed to meet endpoint in the phase 2 study. And what we did is we stepped back, we focused on the data, we dug deep to understand the patient population, dug deep to understand the underlying mechanistic data, and redesigned a trial that was a big success. So I think what I would encourage folks to do is think about how you can de-risk your program. And typically, particularly in biotech, much of that comes through generating good quality data that you can trust, and thinking importantly about, as Michael just said, patient populations.
Right, and this is Catherine. I would like to bounce back on both of you, because I did fail a phase 3 study with a comparator. However, we had a signal, so we went to the FDA and we show the data and we say, we are sorry, but we did not meet the primary endpoint. However, we have a signal. And, for example, could we analyze now the data as a single arm versus, a controlled study? And we are still discussing with the FDA, but they asked us to go ahead and reanalyze the data as a single arm study. So even if there is failure, as both of Michael and Matthew said, there is always maybe either to reanalyze, go and talk to the FDA and explain what you would like to do, your approach, get their feedback. Most of the time, my experience with the FDA is they are willing to work with you because our patients are in need of new medicine. And so they will definitely try to do their best to see if they could accommodate you.
That's
Perhaps
great.
just one more, just brief comment on that. That's a great example. So think carefully, not just about your primary outcome measure. Make sure there are good secondary outcome measures, measures
Correct.
of biological response, et cetera. Think about those just very, very carefully.
Definitely.
Javad, I think your microphone is muted.
Sorry, I'm still getting used to the challenges of online presentation. So yeah, this is really a very good question, and I want to go back to this concept of this reverse thought process. What is it that let's imagine what would be something that we learned during clinical development that we would just say only if we knew that a few years ago. And so one of the challenges that you have in the in development of a product, especially for vaccines, is that the moment you go into the clinic, your epidemiology pretty much goes down the drain. Everything you knew about epidemiology from all the papers that you have read did not have your case definition in mind. They were just general on that particular type of disease, very vaguely defined. So we had a lot of discussion about it, that this can actually, that could be a big threat. I think this happened exactly to Pfizer's, with, they expected a very high attack rate, relatively high, like 1.5%. That would give them the power to really do a phase 3. And once they were there, the attack rate was probably 0.5, 0.6%. So that's just one aspect of it. There's so much to it. So we had a lot of discussion with our advisors here, and then say, like, start today an epidemiology study, which is focused on your case definition. Only look at those people that you would actually recruit in your clinical trial. And in that population, see what's happening and see if there you can find, for example, a correlative immunity there. And this is exactly what we are trying to do now. We're working with a few organizations and also academic labs to get us there. That is extremely important. Also, the other point is about the risk. Just as it was mentioned here, that think about other indications. For example, and you can start thinking about that in your preclinical development. For example, our vaccine, we have tested it in prosthetic joint infections. Doesn't work. It doesn't prevent a prosthetic joint infection, but it did reduce the amount of pus. So I said, maybe we can actually combine this with something else and then do it. So that creates a different venue for product development toward a different indication. Or maybe we can just say that, well, if you have this vaccine and then you use the antibiotics, you have a better clinical outcome. These are all thought processes that provide additional alternative clinical development pathways. We are even thinking about some kind of therapeutic application of the vaccine for atopic dermatitis, because there is a lot of role for Staph aureus in that. So with that, I'll stop.
Thank you, Javad. David, anything in particular that you would like to add on that question?
So, fail early makes a lot of sense, and it's quite a bit easier in the diagnostic space. We don't have to do an IND to be able to do clinical samples, but testing clinical samples as soon as you can is so important in two key ways. First of all, no matter what you try to do to spike samples, you can't replicate what the sick or patient population is going to look like, and so you need to get to see what that looks like. And you see things like what we saw, a higher level of polymicrobial infections than what you might see in the literature, for example. The other thing that is important for a diagnostic is that every hospital or every clinical site is different, and so going through the motions with them before you do a clinical study might alert you to the fact that the policies at one site may not give you or may not treat your sample as it should be. So both in terms of the patients and in terms of learning how the sites work, it's so crucial to do that as soon as you possibly can.
Yeah, thank you. Okay, let's see, a lot of questions coming in here. Some are very program specific. So let's see, okay, for Matt and Chris, here's a question. The microbiome FMT area has seen adverse events due to, in hindsight, insufficient QC/QA of CMC. You guys have multiple products at various stages of development, and so how have these events over the years affected your discussions and your strategy around safety with the FDA?
Yeah, thanks for the question. It's a great question, actually a really important one. Series since inception has actually put patient safety at the forefront, and core to our belief as a technology was, while there was interesting proof of concept developing around the use of fecal microbial transplants, for those of you who don't know what FMT is, and the ability to treat and have an impact on disease outcomes, we said there's got to be a better way, right? So really our products, what we do is we work to identify what the key active bacteria are that are important in the particular disease setting that we're looking at. So for example, in the context of our SEER-155 program supported by CARB-X, we are identified key bacteria that prevent infection, particularly by CRE and VRE, and then as well have in the composition bacteria that we know can modulate host immunity via the gut, that we anticipate will have systemic effects on receptivity to graft versus host. And so in terms of thinking about those quality controls, it's very, very important. As the person asking the question noted, FMT, there have been deaths due to FMT over the past couple of years, and FDA alerts associated with that. And in addition, take your current pandemic. Things like FMT rely on donor screening, and donor screening alone can only detect the things that you can detect and know are there. You can't detect emerging pathogens. And we know, for example, SARS-CoV-2 is transmitted through feces. So, and so what we have done at Seres is built into our manufacturing processes are actual inactivation steps as well that demonstrate that we are inactivating key bacterial pathogens, viruses, fungi, and those kinds of data are part of our INDs for our various drugs. We've worked closely with the FDA to define product identity, purity, and potency assays to ensure identity of the product, consistency of our product, and potency. And yes, those need to be conducted with a high level of rigor, which means qualification and validation. And so folks understand too, those metrics for qualification and validation get stricter and stricter as you move to later stage trials. And so there's a fair amount of work that has to go into that.
Yeah, really interesting. I was going to ask about SARS-CoV-2, both to you and to David, in terms of impact on obtaining clinical samples for screening.
Sure. Well, at Seres, we have two manufacturing processes for our products, and it depends. We have biologically sourced products which fractionate and purify the bacteria from biological material, and for that we have stood up our donor operations to be able to collect those biological materials and have important safeguards in place with respect to that. But importantly, again, as I just said, our manufacturing process then inactivates anything that might have gotten through. But I can tell you, it's pretty hard to be a donor in our programs. And of course, we also monitor people consistently throughout the process. And then products like SER-155 that CARB-X supports are manufactured by cultivation of strains from our strain library. And so in both of those cases, though, we pay particular attention, obviously, to the safety parameters, and have worked with the agency to define those.
And I'll just add that doesn't stop with our CMC release measures, as Matt was just describing, right? The FDA in our pre-IND discussions with SER-155 was very keenly interested to understand not only those measures, but how we're going to monitor safety in the trial. And I think that touches back on that, like, fail early type model. You got to walk before you can run. And so we're really structured that clinical trial around this idea of patient safety is the first thing we need to assess, particularly in vulnerable patient populations, but really across the board. And so we're taking a very measured approach initially as we do first-in-man studies to understand the safety of the drug and monitor that very closely. And then as we get that more full picture from the clinic, we can modify that design as we go.
For us at HelixBind, the pandemic has presented quite a bit of challenge for our clinical studies. Many of our sites shut down studies because they really need to focus. So that's been quite problematic because we learn a lot from our sites. On the other hand, there is an opportunity for us that we're somewhat excited, and that is, for good or for ill, many patients, many COVID patients, or a majority of COVID patients are on empiric antibiotics because clinicians are going to do whatever they can, and there are signs of sepsis. And as I mentioned in my presentation, once the patients are on antibiotics, culture really is of no use. And so our test is perfect for that application. And so there are a number of investigators who are excited to look at that, particularly in COVID patients with us.
Very interesting. Okay, let's see. Here's one for IBT. Let's see. Well, there are two, but I'll start with this one. Staph aureus has made many superantigens, but your vaccine has only one, probably engineered. Do you think that removal of superantigens would make an equally effective vaccine?
Okay. That's a very good question. First, I have to clarify, it's not only one. We have actually a fusion protein of 3 superantigens. So just for the background, there are about 20-some superantigens that Staph aureus produces, but not equally frequently and also not equally potent. So what we did is that we actually looked in the literature first and see what are the most prevalent ones. So we picked the most prevalent ones, and then most prevalent prior to USA 300, which is the community acquired. So the USA 300 strain has a different one, which is called SEK. We chose SCA, SCB, and TSSC1. And then we actually went through a lot of human plasma and looked at and then purified antibodies against these different superantigens. And then we saw that the combination of the antibodies against three superantigens actually provides a fairly good broad response to many of the other superantigens as well. It doesn't cover all the 20, but it does cover the most important ones. And therefore, that was the strategy we took.
Very helpful. Okay, this is for your colleague, Catherine. Catherine, IBT-V02 is a complex vaccine. Did you have to present preclinical protection data for each individual antigen, justifying the inclusion of each in the vaccine?
No, we did not.
No.
Because we did not present the vaccine as a combination. I mean, basically, when we went to the FDA, we did not say each of those are active ingredient. So it was not a combination product. It was, these are the ingredients. and this was the active ingredient. So if I remember properly, and Javad, you could correct me, we had some discussion with the FDA regarding the fact, or maybe it was with the European agency, saying that we needed to do individual to show individual protection, and we took them out. So we did not.
Yeah.
Okay. Thank
Actually,
you.
to this point, there is a really important thing that, what is your product is the question. You take one of them out, that's not the product anymore. In fact, a lot of people have, in their vaccines, alum as an adjuvant. We found out that you need alum in order to have stability. So therefore, before considering alum as an adjuvant, it's an indispensable part of the vaccine. Therefore, without alum, there is no product. That simplifies your clinical trial because you don't need an arm with adjuvant only, because we don't even call it adjuvant. It's an excipient.
Right. Yeah, that's interesting. Good point there. Okay, this is for Bala. Bala, you had mentioned, in your slide where you gave different perspectives from, Bugworks perspective, the grant agency, and the investor. And so a question that's come in, which is, what would you do differently to obtain better guidance from the FDA?
Well, we have to submit the IND in a short answer, because a pre-IND, if you remember, does not have the reports going along with it. It's a synopsis document. And one of the key things about the complete feedback to expect from the FDA is when they go through the detailed reports, because there's always fine print. So based on the briefing document, which is what is required for a pre-IND, that's the level of, it's sort of top level. And to get more feedback, you actually have to go through an IND submission with all the tox reports, with all of the preclinical reports. And that's when you actually get a binding feedback in terms of, in terms of the clinical progression.
Okay, great. Michael, quick question for you. Do the Amicidins work on biofilms?
Actually, the beta is designed specifically to be helpful with biofilms. So the alpha is more of a barrier product. The beta is one that has great antimicrobial activity, but is also designed to be more aggressive. It's a surfactant or a cleansing product to help disrupt that biofilm.
Okay, great. Thanks. Okay, back to Matt and Chris. I guess this is a two-part question. What is the duration of treatment is part one, and then part two, how does the human gut microbiome vary across the world?
Sure. So I'll lead off, and then I can flip to Chris as well to add anything. So the duration of treatment for our drugs really depends on the disease indication. So in the context of our C. difficile program, it's an acute infectious disease. And so we're looking to rapidly restructure the microbiome and prevent both the germination of C. difficile spores and the vegetative growth of those bacteria. And so we seek to achieve engraftment of our bacteria very rapidly, and we use a short acute dose, which is dosed over 3 days, is the dose regimen. And what we see there is that our bacteria onboard very, very quickly and change the metabolic landscape of the gut to be unfavorable to C. diff. And there are two primary MOAs in that case. One is the bacteria in our drug can compete with C. difficile for important carbon sources, in particular certain amino acids that C. diff favors in an antibiotic treated gut. And then as well, secondary converts primary bile acids to secondary bile acids and restores that capability. And bile acids play an important role both in C. diff germination and vegetative growth. For our chronic diseases, for example, in the context of our inflammatory disease and our work in ulcerative colitis, we actually dose over an 8-week period of time, and that helps both, again, rapidly restructure dosing early on, but then to maintain a reasonable level of the bacteria. That is something that we continue to evaluate as a company, what the optimal dosing regimens are, and that's ongoing work that we have. I'll point out what we have observed across our clinical portfolio to date in human subjects is that we do get durable responses. So once the bacteria are onboarded, we do detect them once we cease dosing. And Chris, do you want to take the geography question? Go ahead.
Sure, it's a great question, and one that we're sort of constantly keeping an eye on as you continue to see new population level surveys cropping up across different geographies. The short answer is that while there's a lot of species to species variability, by and large the functional capacity of any given healthy microbiome tends to be pretty constant across geographies, and so there are sometimes different bacteria providing those functions in different geographies. And certainly geographies that are more mixed or share similar lifestyles tend to be more similar to one another. But the functional cohort there tends to be pretty consistent. I think there are some interesting examples cropping up across the literature of specialized functions that often relate back to diet or lifestyle. Those tend to be the exception, not the norm. So there are nice little case studies or nice little examples of particular metabolic pathways that are related to the intake of seaweed or other sources of or other differences in diet. But again, by and large, there's more consistency than there is difference at that level. I think one of the challenges in this space, and it's one of the things that we face even on our clinical trials, is how do you standardize these types of data such that they're readily comparable, right? So the advantage we have at Series is all of the data that we generate in-house comes under the same pipeline and same umbrella, and so we're able to use that to rapidly translate our clinical trial observations into new product development. But when you pull in data from external collaborators or from public data sets, you have to start by understanding the fundamental technologies and methods that went into that data generation, because those by themselves can drive some of the differences that have been observed when comparing these data sets. Matt, I don't know if you have anything else you want to add to that.
You've covered it. Thanks.
Okay, and I'm told that I need to start closing, but a few more questions, quick ones. David, question for you. Can your instrument be used for point of care testing?
Technically, yes. We will have to go and get CLIA waiver once we have FDA clearance to support that, but technically, yes.
Okay, great. And one last question for you. How do you define, quote, relevant load, unquote?
That's a good question. In sepsis, no one really knows what the relevant load, and if you talk to one microbiologist, they say one thing and another. We can, in fact, get a measure of load, so we will know that, and we're finding that out during our clinical study. But in terms of marketing, you really need to get down to the single CFUs per ml as a product.
Helpful. And now I see Astrid's popped back up, so I think this is definitely time to start closing down. But I want to thank all of you. Really, really great presentations. I think gave us a really interesting view across all these modalities and verticals, of approaches to AMR and what you need to do in terms of engaging the regulators and shaping your program. So thanks a lot. Really great insights. And Astrid, thank you for all your hard work and setting up this webinar, and certainly to all of the attendees. I'm sorry we didn't get to all of your questions, but I think we covered many of them.
All right. Thanks a lot, Erin, for moderating the Q&A and also, of course, for putting the panel together. And of course, a big thank you to all our speakers.
And Erin and Astrid, thank you very much. Great meeting. Nice to bring these talks together. I really enjoyed the talks. Thank you.
Well, now you can go have your breakfast, Michael, and Bala can go have a late dinner. So.
Yeah, there we go.
Thank you all. Thank you very much.
All right. I just want to still take one moment, at least for our audience, to announce our future webinars. We had a little low time over the summer, but we are now coming back in full force with our webinar series. On the 8th of October, we have another webinar from which is also coming out from the bootcamp series, which was supposed to be for the ASM ESCMID conference. It will be on exploring safety issues in antimicrobial drug development. On the 21st of October, we will have a webinar about data and methods needed to determine breakpoints for new agents. On the 27th of October, we will have a webinar about the medical need and the regulatory challenges for developing antibiotics for children. And on the 17th of November, there will be a webinar on the discovery of new antibacterials using artificial intelligence. So you can already register for most of the webinars on the Revive website. Those who are not there yet, which are not there yet, will come in the next days. I also recommend to you to keep an eye out for our emails, because there will likely be other webinars before the end of the year. And finally, if for everybody who registered for a webinar in the past and has not opted out from our communication, but still doesn't receive the emails, please reach out to me and we will figure out how to get you properly on our mailing list. All right, so this is really all now. It's all from my side. So again, thank you to everybody, to the speakers, to Erin, and to our audience for contributing to the discussion. I hope everybody found it useful and interesting, and that you will join the future webinars. And also make sure to spread the word and encourage your colleagues to join as well. Thank you, everybody, and goodbye.
Thank you.
Thank you again. Bye now.
Thank you.